Okay, so this is much less of a statistical view of R and those kind of things, and much more a pragmatic view of how you can use some of these multivariate statistics. So I did do statistics at an undergraduate level. I can derive these things from first principles, and I can tell you, even to my math-loving heart, it's a pretty boring, stupid thing to do. So I'm actually just going to step through a bit more lively way of thinking about some of these things. So multi-dimensional data is really hard for humans to think about. We can cope to about 3 dimensions and our brain goes, I wasn't made for this. I'm going to start bleeding out your ears. So on the indicator side of thing, or a model side of thing, you can be presented with thousands of indexes or thousands of variables that you have to think about and you have to combine. So multi-dimensional, multivariate analysis is a way of finding your way through that space. Most of them can be used to explore your data. So some of the questions that you were posing to Scott before, you can use these to explore your data, but you can also use them to confirm hypotheses you have about structure you think's in that data. I'm going to concentrate on the first 2, but they're all kind of connected. And then Scott's going to talk about the last one in a little while. So the key part of all this is data is just the information, the structure, and the noise. Okay, and all these multivariate methods are doing is helping you pan for gold. You're getting your information and you're chucking out the dirt, the noise that you don't want. So if we start with cluster analysis, this is about, in that high dimensional space, each one of these dots represents a data point or a sampling site or whatever you're looking at. You're trying to minimize the differences within a cluster and maximize the distance between clusters. So you're trying to find natural lumps that are in that data. There's lots of different steps to go through. The first one is to prepare your data. The number of times I've had a student come along and say, look, this plot data just doesn't have any structure at all, and they've still got all their missing values in there. You need to make sure that you've gone through and tidied up your data first. You know, always eyeball your data first, always go through it and just make sure that it actually is clean and sensible and there's no really obvious typos in there, for instance. I have a student who couldn't figure out why his data analysis and his model wasn't working. It's because he had a shark that weighed more than the entire mass of the universe because he'd missed the plus sign in his E calculation. So, you know, you have to check. Then you have to pick which distance metric you're going to use, so how you're going to measure the difference between the clusters, and I'll go through what those metrics are in a minute. Then how you're going to check whether those clusters are really separate. What's the linkage criterion? You know, how are they being linked or pulled apart? The actual algorithm that's used by the calculation. And then whether you believe the clusters that you get out the other end. Do they actually make logical sense? Never write a paper, whether it's a modelling paper or a data paper, that says, because the computer says so. Okay, I can't tell you again how many papers I get that says when I've got a model, but it balanced. Yes, doesn't mean it's right. Okay, so you have to make sure that it makes sense. So if we look at the distance metrics, the most simplest one, the one that we'll be most familiar with from primary school, is just the straight line between 2 points, the Euclidean distance. That's the simplest measure to use in all of these multivariate analyses. The second most common one is called the Manhattan City Block. It could have been the Shanghai City Block or the New Delhi City Block, but it's, think about it as a person walking along one street and then turns and walks on the next street. So these are the different dimensions of the data. And all it does is it's calculating these distance. And it does it over all of the different dimensions and then gets an average. So this is just an average straight line distance, and then this is an average stepping through the dimensions. And they're the 2 most common ways of looking at distance. There's a whole bunch of other ones that you can then get into that are built on the Manhattan idea. So I can't say most of these names. I think you say that's Chebyshev. All it does is it doesn't do the average, it just finds the biggest distance in all those dimensions. Another one is the power, where it takes again this idea, but it weights it for the different dimensions. You might want to have the employment dimension weighted more heavily than the biological dimension, if you're Rashid, for instance. So that's where you can weight your different dimensions. But you can also weight it on uncertainty or the pedigree of where you've got the information from, whether you've directly collected it for the region that you're really in, or whether you've taken some global national level database as a proxy. Most of the time you see a discussion around these methods for continuous data, but you can actually do it on categorical data too. So the distance measure in this case is just say you've got a ranking system from 0 to 5. Okay, so if the two data points in the different dimensions share the same score, they match. If they don't, then they don't match. And then you just average the number of matches across all of the dimensions, and that's your distance measure for categorical data. The next part is the linkage rule. So how do you tell if data points are in the same cluster or how different those clusters are? So again, the most simple one is just the nearest neighbours. How close are you to your neighbour data point in that multidimensional space? Okay, so that's the simplest one. The other one is called complete linkage, and it just reverses that idea. It says, okay, what's the most distant one? If I've got two clusters of data, what's their most extreme difference? There's a whole bunch of other ones then that looks at pairing up points in the two different clusters, whether you weight that distance or not. There's ones that also take the centroids, the centres of gravity of the different clusters, and looks at the difference between those, weighted and unweighted. Most of them, though, are actually, except for the simplest one can kind of work regardless. Most of the fancier ones that statisticians get all excited about only really work on really lumpy data, where there's really clear differences between the cluster types. The one that most machine learning people get really excited about is called Ward's statistic, and it's really just doing an ANOVA in multidimensional space. It's finding the minimum of the sum of squares between your different clusters, which is really computationally efficient and useful for some of the face recognition stuff they want to use it for. But it creates quite a lot of small clusters. So for someone who's trying to get big ideas about how the ecosystem's working or how the socio-ecological system's working, it can actually not help very much. So then you get on to the algorithms that you want to use. So there's two different major divisions in the algorithms. One where you think there's actually like a hierarchy of nesting of the different clusters, and one where you think there's just clusters and there's no extra structure to how they're connected. So if we think about the non-hierarchical one first, the main way that people use is called the k-means method. And all it's doing is it's saying, look, there's a massive data points. Are they around one particular centroid? And how many centroids are there that they could be clustered around? The biggest drawback to doing that is that you have to guess how many centroids there are in the first place, which seems a little bit circular to me. I'm not an expert in this one though, so there could be people out there who are quite happy using it who should speak up and say, no, it makes sense because of this reason. And the other ones, one is again has that centroid idea, but it assumes all the points in each cluster are drawn from a Gaussian normal or some other distribution. So it's looking for the minimum number of distribution centroids that would then explain the data you've got. And the last one is just looking at layering up data points. What's the density-based one? So where are the points all on top of each other? That's going to make a cluster. So this one is actually trying to show you that this is from a distributional one. Pictures were just taken off Wikipedia, I must admit. So this is just saying you can see there's some clear clusters here, and even this one, even though it's fairly sparse, can be drawn from a distribution. So all of these ones can cope with this because there's clear lumps that they can find. They're going to have a lot more trouble with this one because there's long smears and no clear difference. So that's where these kind of things can be tripped up. The more and more dimensions you have means that at least in some dimensions you're going to have that smearing effect, which is what's going to undermine some of these and why it gets harder with the more dimensions that you have. The other main way of thinking about cluster analysis, the one we're most used to seeing, is when they create trees. So they're called dendrograms. But basically what it's doing is saying, we've got all our samples together here. Okay, let's start splitting apart those clusters. The one that's most different comes off first. So this could be sort of all life, and then this is bacteria. And then you go to the next lump. Okay, these are things that have bodies that we can see. So then we've got the invertebrates come off from the vertebrates. And you can create a tree by looking at how those clusters step through the nesting. To give you a bit of a tangible example of that, I work on quite large complex ecosystems, both where we're getting input data to think about, but where we're actually trying to interpret the outcome of looking at management strategy evaluation. So that means you play out the management strategy inside a computer model and just compare what worked and what didn't, okay? But you can still be presented with hundreds of different management strategies that you've got to compare for thousands of different parts of the ecosystem. So this is an example where I was trying to figure out what was happening. So I just looked at whether the different classes of animal in the model separated out differently. What we found was the really high value target species separated out from the rest. These two here were habitat dependent valued fisheries or fish species. These two here weren't as high value, and they also didn't care about habitat. So you can see there's starting to be a reasonable understanding of what's going on. These two here were highly mobile things. These things here are habitats. These are prawns. And then the seagrasses and turtles, so the turtles were really dependent on the seagrass, lump out together. So the clusters it came out with made sense. I could see the story that was coming out of that. So it was the way of validating what's going on and really appreciating the structure that was in that data that I just couldn't see when I was presented with, you know, like 10 terabytes of information. Yep. So there's single value decomposition and algorithms. So typically just go to a multivariate statistics textbook, or in this case R. So here you're basically running a single value cluster analysis, and then you plot it, and that gives you your dendrogram, okay? So it depends on how much mathematical information you want, Andre. So I don't think the audience here is actually going to be prepared for me to sit through going through the statistics of it, but you can get quite detailed information about the components, setting up the correlation matrices or covariance matrices that you're creating it off, going through the steps of if you wanted to do like a Python script, I don't know if there's a Python script for principal components or cluster analysis, but if you wanted to write your own, there's plenty of documentation out there in quite thick mathematical textbooks or how-to books that I'm happy to chase up with you afterwards. But this is just an example of the R scripts to pull this kind of stuff together. So at the very start, you have your data set. You want to get rid of any missing data, so you're just omitting them. Then you can rescale them to standardized variables so they're in comparable dimensions. You don't have to do that step, but it actually makes the computations faster and easier because it's not trying to deal with things that are like from 0 to 1 in one dimension and then from minus 10,000 to a million in another dimension. So you're just standardizing everything so it's a bit easier to deal with. Then I've calculated the distance metric. It's just a Euclidean distance metric. So in this case, this is what Scott was talking about before. This is the name of the function here. I'm sending it this data set. And one of the key words it wants to know about is what method I want to use. So again, you just go on, in this case, R. You look up the list of options and you pick the one that you want to use. Okay, so I've just picked the Euclidean distance here. Then you run your clustering analysis and you send that information to this thing so you can plot it later. So this is just running your cluster analysis. It's using it from the distances you've just calculated. And this time I've picked the really simple way of looking, the nearest neighbour way of looking for clusters. Then out the other end, I just plot it. So then you've got the idea of thinking about what structure you think is in there. So you look at it and you say, well, I think there's 5 groups that have come out of that cluster. In this case, 5. So this is a way of cutting up that tree and saying, I think there's 5. Where does the computer think those 5 might be? And then you draw it, you redraw your dendrogram, but this time you draw on where it thinks those clusters are. It puts a nice little box around it. Then you can see whether the computer's interpretation, which is supposedly more objective because it's just running off numbers and it's not being tricked by your eye, agrees with where you thought those clusters were. So this is about the mathematical validation of where you think that structure is, and then you have to go through the process of making sure that you can understand why you think it's grouped it that way. So for the use of indicators for informing management, you have to understand where those signals are coming from. Black box indicators aren't that useful. In the machine learning world, the AI world that runs your computer game, if you're playing a first-person shooter like Ghost Recall or whatever, it doesn't care. It can just use those blindly. But because we have to inform management, it matters. Okay, so the most commonly used multivariate statistics I use is the PCA. So there's a really beautiful one on YouTube that explains PCA is turning a teapot to get the most information about a teapot. I actually learnt about it as pizza, mainly because I had an Italian professor who always had his lectures at lunchtime. So for him, each piece of pepperoni was a data point, okay? So he would present us with the pizza at some odd angle. He'd literally come in with a live steaming pizza that was flopping places, and he'd be holding it up at odd angles and say, how can we get the most information out of this pizza? And we're all just thinking, eat it for God's sake. So what he actually wanted us to do was move it around into a place where you could see the most of the pizza, so you could see what toppings it had. And this picture doesn't do you justice because you have to have it slightly tilted so you can see how deep that pizza pan is, but I couldn't find a good enough picture for that. So it's about transforming the view you have of the data so that you can see the hidden structure. So again, lots of this stuff, if you're not familiar with it, there's plenty of stuff online now. So Principal Components for Dummies, for instance, has all these kind of stuff spelt out quite simply. You're trying to find the axis through the data where there's the most resolution between those data points. So if you put a vertical axis here, these data points are still pretty tightly clumped on that vertical axis, so you're not getting enough difference between them to see that structure. So you're trying to maximize the variance that you're seeing across the data points. However, if you put a line the other way, you have got the most spacing that you can get through that. So that's actually what you're after. You're after maximizing the variance. So when you talk to the mathematicians who get excited about this, they talk about the eigenvalue and the eigenvector. Just to interpret that for an ecologist in the room, the eigenvector is the direction of the line, and the eigenvalue is just the amount of variance that you've described. Okay? The other thing to remember is that you're not really doing anything to the data. So just by doing a PCA, you're not, in the very first step, reducing the number of dimensions it's got. So if you've got n-dimensional data, so just say you've got 3D data, it just rotates that. So you're going to end up with 3 eigenvalues and 3 eigenvectors, because it's really just rotation. So the reason to do that is to try and make patterns clear, though. So if this is our original data, and we tried to plot it up and then use straight statistics on it, the red one would have that kind of distribution of points and the blue one would have that. So most numerical packages would say, actually, there's no difference in there, okay? But there is some underlying structure that if you run the PCA, so you take your axes and you rotate, it's now much clearer that you can see that there's some clear structure falling out there. So it's typically they're not nicely coloured points ahead of time, so having that moment of, oh yeah, there's a pattern is a really cool feeling. Or maybe I'm just weird. The part about it is that keeping the important bits, and the bit that can really trip people up is that these principal components, as they're called, the new axes, they don't have a real world meaning. Okay, they're just mathematical inventions. So you've got to then try and reinterpret them based on a variable that you actually understand. So that's when you plot out the variable, so that the properties of the system that you're thinking about, to see what properties were influencing which axis. So this axis here is being influenced by temperature, so this one along here, whereas this axis here is being influenced by effort. So you can do and see what's drawing out those different components. The axis, which variables were influencing those components the most. Kind of linear. It gets a little bit difficult at that point. But yes, they are combinations, but they don't necessarily... I mean, you can have a case where all of these influence guys are like completely at 45 degrees and, so it's not necessarily a clearly... Very rarely do you nicely get temperature has now become X and effort has become Y. Okay, so you really have to do the... I'll show you an example in a second, but you really do have to do this step to understand why those clusters are being pulled apart. So to get through these steps, it uses the covariance matrix. So this is just, again, doing what Scott did before. We're just reading in a table from a CSV file. I'm telling it that it's separated by commas. Sometimes there can be spaces. In this case, I've got a header. So I've written in names across the top of the file of what they are. So I'm just telling it that that first row isn't important. That's a row. It doesn't have to read in. That way you don't have problems with that. Then in here, I'm just saying, okay, we want you just to recognise that I have some column headings that I need to worry about down the track for some plotting reasons. Then it tells you I wanted to pull out some particular parts of a huge data set. So you don't have to use every column in your data set either. You can come along and tell it which columns that you actually want to use. You can tell it whether it's the covariance or the correlation matrix that you want to use in your PCA. In this case, we've used the correlation matrix. And then just the data that you're actually putting it in. And then you can see, to answer your question just before, you can see what the loadings are for each axis versus each of these things that you've told it that you want to think about. I don't actually have a plot of that table there, but it just pops up and it's a matrix of, for each of the properties that you've put in, how that loads across the different principal components. So you can either do that in table form or plot form, depending on whether you're a number person or a visual person. The key part to worry about when you're looking at PCA is to understand the amount of variance explained. So this was a snapshot from a very long table where I had 12 dimensions I was worried about. You can see the cumulative proportion of variance of the first principal component here is only 30% or 25% effectively. That was really disappointing. I can't tell you how disappointing that is. You're aiming for like 80 or 90 here. That's a good day. When you get really low ones, it means you've got to consider even more principal components because you're trying to explain as much of the variance as possible. So I've cut it at 60% here. In reality, I probably should have gone a couple more steps to 80%. You're trying to explain the majority of the variance in your new space. So you plot that up. So the easiest way of looking at it for me was to plot it up and say, what's the cumulative amount of variance that's left? So in this case, I should really be coming down to 3 or 4 principal components before I say I've explained most of the variance in my clusters, my groupings. What you're really hoping to see is something like this, where, yes, I don't have to worry about most of the dimensions because there's just a couple of dimensions that explain it all. It's not always that easy. So this is a real-life example. So we've decided on how many principal components to keep. It's kind of hard to plot more than 3, and most people only plot 2 at a time. You can plot different principal components, like go 1, 2, 1, 3, 1, 4. That is a way of piecing it together. But most people plot 1 or 2 because we can cope with that. Then I've put on the loading vectors. So these is some real data points that I've used the numbers because they were the codes for certain outcomes, certain scenarios. And then I've put on what was influencing those, which you can't really see on that plot, but you'll be able to see on the next one. So that's just a biplot. The other thing I should have said was that there's many different ways of doing a PCA in R. There's a couple of mainly used functions that some of them do plotting a bit better than other ones. So this is an example where it's the same, slightly different data set, the same idea. What I've done now is I've come back across and just myself, because I know what these numbers stood for, grouped them up. So this is a case where we're looking at management strategy in Australia under different climate scenarios. What we can see from this was that biodiversity and value were pulling out this one, where it was plankton productivity and pelagic food webs were pulling out the top ones. So you can't read it here, but that's what it says on my computer. The screen resolution is a bit too low, unfortunately. And then what's happened is that because I can put on the codes of what these different data points are, I can see that it's the low emission scenarios have grouped out together at the top. The intermediate ones have happened in the middle, and the high emission scenarios have come out here. And a case where I wasn't allowing for evolution of species has fallen out completely by itself. So something that was an impenetrable mass of outcomes in a spreadsheet for me suddenly got a structure that makes sense and can help me take a few more steps. Other people plot it up in a different way, which is to look to see what their different factor types are doing versus the principal components. So you can see that there are the different structures coming through. So that's another way of doing those red arrows and loadings as well. Yep. In this one here. This is a picture I just grabbed off the internet because I couldn't be getting it to work on my data. But basically what's happening is this is your variable, and this is the loading for that particular principal component. Okay, so it's just saying that for this variable 1, there was like a 0.18 loading for this principal component, whereas this variable here had a minus 0.2. So it's how the loading is, what that translation has been for that data point. The last step that you should, yep.
You said that sometimes you use covariance and sometimes correlation. When do you use which one?
Typically, I tend to use correlation the whole time. That's, again, something to chase up with a proper statistician. It's been 20 years since I did stats as an undergraduate, but we can look into that this afternoon together if you'd like. But yeah, it's always good to get a real statistician's advice. I tend to use these as an eyeball method as much as anything, but you should always double check with the experts. One key part of doing these that typically is forgotten by most people is to do the equivalent of a residuals check. So if you do a regression or a fitted model, you look at your residuals to see if there's a pattern in the residuals. Most people forget to do that in multivariate space. In fact, it's really hard. Well, it's not straightforward to do in R. You actually have to write it yourself a little loop. It doesn't fall out of any of the PCA functions that I could find very easily. But what this is saying is that most of the data points are actually well leveraged. They're all on top of each other. You've got these out here that look like outliers. They're sitting out wide. If this was a real data set, you'd want to understand why those outliers are there. Should they be omitted? Is there a problem with them? For me, it's actually fine, because I know that these are actually all the ones where I hadn't allowed for evolution. So they should be different. They should not have the same leverage on the stuff as what was happening here. But it does mean that they're not being as well explained in that higher dimensional space rotation as your other data points. So it's always good to check your residuals. Okay, so the last, I won't go into as much detail. Yep. Oh, sure, go for it. Okay. Names. Yeah, no, that's cool. Cool. No, that's cool. You can put time series in it, yeah. You can put anything you like in it. Yeah, so you've got to have a column in there to tell you it's time, probably. You know, it's set up like any other data set. You have your distinguishing row markers, and then you have your different columns of data. So it can be time series, it can be snapshots from different locations, it can be, you know, the stuff that you guys are doing where you're getting multiple indices, either at different locations or through time. You can put all that through a PCA. Yeah. There was another hand somewhere. Okay. No, go Scott, that's okay. So there is quite a lot of discussion about very fine details in the stats, to be honest. But we'll get to that in a second. Yep. No, the rotation all comes from the PCA itself. Um,
But I always have the problem that I really don't know which kind of version to use at the end, so it's a little bit tricky for the parents to I wonder if you have any kind of
no, to be honest, if I suck it and see. If it's still not making sense, I go and see a statistician. But yeah. There's a question down the front. Did you have a question?
Even when you do cluster analysis at times, there's some, after you've done the statistics or the analysis, there's some who recommend you use doing a confirmatory test or a t-test, if they say. Like even after you see clear clusters, how important are they and how well can you do them in?
So that's again comes down, well, you can do t-tests in R pretty straightforwardly. But the standards that you go through is depending on how you're using it. So for me, it's an exploratory tool. It's not a so I use it as an exploration tool to help me visualize the data, okay? So the statistician who taught me basically said, if you can't see it in the data, it's not really there, okay? So lots of statistics tests will say there's a significant difference just because there's lots of data points and really there's no real difference there at all. So if you can't see a difference, it's very unlikely to be there. So that's what I'm using these methods for, to have a look at the data. If you're using it to confirm a hypothesis, which is another way of using these tools, then there are extra steps that people, by convention, like you to do. So t-test is one of those. I don't have as much experience with that because it's not the way that I use the tool.
Clear clusters which you can clearly see.
Yeah.
Do we still need to do a t-test?
Again, it comes down to what's the convention for your discipline. So in the marine ecology world, you wouldn't need to. It might not be the same in geology or molecular biology or something. So that's kind of, you have to go back to your discipline and find out what's appropriate there. Yeah. So the couple more that are related, again, in the statistics, so to an ecologist or to a pure mathematician, the maths is the same. So the same kind of point as Eric was making before. To the people within the stats world, they do get into the kind of conversations that Scott highlighted before, which is why you do need to go to a statistician to make sure that there's not a subtlety that you've missed. But basically, if you look at factor analysis, it's quite similar to doing a PCA. The main difference is what you're looking at. So in a PCA, you're looking at total variance in your data. In a factor analysis, you're looking at the shared variance. So not the novel parts, but the shared variance that's inside that data. It's really just looking for latent underlying structure that's in that data. So most of the time it's used for an exploratory data use. You can, however, use it to create that hypothesis testing we just talked about there, where you have a training set where you think you've set up clusters, and then you have your test set to see if you can take an observation and get it in the right spot. If you explain what's going on. And that's actually even more used for canonical discriminant analysis. This time it's actually looking for correlation structures rather than the variance structures that's going on, so linear combinations. And it is really trying to get a set of linear rules to apply. So if you're given an observation, it takes the rules and said, right, you will end up in that cluster. It can be useful for filtering data or finding structures, but it's mostly used in face recognition software that you would have gone through at the airport, for the marketing that targets you if you get rung up at home with those spot calls, and also for credit scores in the United States. So it's all about, it's used in other parts of society, and that's why they want these simple rules that help you classify things. The last part is over to Scott again. So it's a multidimensional stuff, and we'll get Scott to follow up on that. So while we swap headphones, is there any other questions? Do you want the fancy stuff? So this microphone here's on the outside.
Okay. Okay, so I'm just going to briefly go over non-discriminant multidimensional, non-metric multidimensional scaling. Yeah. So it's an ordination technique, a little bit different from PCA and some of the other ones that that Beth went over. And instead of calculating a bunch of different axes straight up, it actually, you have to specify how many axes you're interested in. And so you explicitly choose how many combinations you're interested in beforehand. And so it iteratively goes through and moves the data around to make it fit to that number of pre-specified groupings. So after a solution is found, it stops, or it just won't run anymore. So it does not identify a unique solution every time. So it goes through this iterative process to find the solution. If you started at a different point, it could come to a different solution. So it's a little bit different in that way. And it's also not an eigenvalue vector technique like PCA. And so your axes, your PCA or your axes aren't really the same. So with those sorts of caveats, why would you even want to use it, right? So you don't really have to make as many assumptions about the data. You can use many different data types, and most any distance metric can be used with your in non-metric multidimensional scaling. And it also pairs really nicely with clustering. So if you wanted to go through and cluster something beforehand, the non-metric multidimensional scaling is a good way to go and sort of verify that. So a couple of pitfalls with MDS is it can be really slow if you have large data sets because it is this iterative process. And it requires optimization, so you can get stuck in local minima, and in which case it won't really converge and you'll have to start over. Yeah? I'll discuss that a little bit, but no, I don't know about that. So basically how it works is you have a matrix that you have laid out where you have your samples as columns and your rows, or samples as rows and your variables as columns. And it calculates all of the pairwise distances between all of the variables, and when you specify the number of dimensions, which is m, it starts with an initial configuration, and then it measures what is called stress, and so it measures how much a new configuration improves the fit based on the original configuration. And so it wiggles everything a little bit, and then it measures the stress, and then wiggles everything a little bit more, and so it iteratively finds the best solution. So then the original distances are regressed against this initial configuration, and so then the stressed is the sum of squared residuals of that process. So basically you start with your set number of axes, and then it goes through that. And so to verify if we have the correct number of predefined dimensions, you can do what's called a scree plot. So you can look at your stress in response to multiple dimensions. So you say you could start with 5 and then look at 4 dimensions, 3 dimensions, 2 dimensions. And then you can look at the stress associated with that. And once you have an area where you're able to minimize that stress, then you have a pretty good idea of a good fit. And so another good thing is to make sure that the solution actually makes sense. Because if it is telling you that, something that shouldn't be, you should probably start the iterative process over again. Which is sort of a funky thing about this is that you can actually get different answers every time you run it. So. Right, and so one of the R packages that is commonly used for non-metric multidimensional scaling is called meta-MDS, and it actually has a little bit of a function where it assigns the axes in a similar way with a PCA. So you can actually look at the axis number 1 as the one that explains the most variance. So here's a quick example in R. So the mass library holds this function called ISO-MDS, and meta-MDS, the one that I was just talking about, actually uses ISO-MDS as a function within it. And so a lot of the R data, a lot of the R functions actually include data sets within them. So here, this vegan package actually has this ecological data set called Dune. And so that's a really common way to sort of test out a new function, is to use the included information. So this is a sample from a run that I did. And so basically you can see that the first run, the stress was 0.05082. And then the second run, it actually increased the stress a little bit. And then the third run increased the stress a little bit more. And so each one of those, the computer was shifting the points around a little bit. And then eventually it comes to the best solution. And this is the output here. It's kind of small, but the red bits are the sample sites, or the actual rows, and then the bigger dots are the columns. So that doesn't really help out too much, right? So there's a couple of other ways that you can make these graphics a little bit more helpful. And so another good thing to do is so you take your previous best solution and then you run it again and see if it comes up with an even better solution. So this is an example of that. And so it actually has higher stress, so it doesn't really look like it's helping out too much. And so there's another example of how you can plot. Again, each of those red labels is a site location, or is a sample, and then the numbers are locations. So, like I said earlier, it pairs really nicely with clustering. And so this is exactly the same code that Beth mentioned a little bit earlier, using the hierarchical clustering. So what you do is you just see how your hierarchical clustering finds the data, and then using multidimensional scaling, you can actually highlight those clusters. So here, these are little trees that are pruned to show the clusters from the from the cluster analysis. Yeah.
Why right now I understand this NMDS as like hard coding or trial and error type of, but its objective is also the objective of PCA. So why use this? Is it you use this when PCA doesn't give you a logical solution? Is that it?
I think that its strength really comes from being able to use different types of data. So if you, like I know, Alida, you all used it for the fisheries biology paper, the diet. And why did you pick it overtly over PCA? Or was it just...
It doesn't make any assumptions about distribution. I judge that the PCA data should have a normal distribution. So NDS doesn't make those kind of assumptions. And if I remember correctly, it also handles basic zeros, whereas PCA has...
Okay.
So
Yeah, those are two big things.
But for some statisticians, they feel that PCA is a more rigorous approach. And I think that's really a matter of personal opinion as opposed to hard advances and better than that. I think it has slightly different use. It really depends on the type of data you have and the questions you ask.
Can have to do with PCA. As long as you have no mean. As long as you remove the mean, just that the mean is zero. Then the zero is one.
So if you center your data.
Yeah. That's
But the kind of data that we were using, which was some data, was Scotch Covariance, and that's had a lot of zeros. So you have a matrix, these are all
So.
If you have community data, for example, and it's very organization, and if you PCA, very strange. Well, if you PCA from parameters, it's much nicer. So then you can run yes. And I want to ask, how is the MDS, this meta MDS, different from, correspondence analysis?
The canonical correspondence, that's I think that's a little bit different. I mean, they all have, it's the same idea, but I can't answer exactly why those two are different. Yeah. Does anybody know canonical?
So it's some of the metrics so it's the statistics that you're doing it on, whether it's variance or correlation, or it's what it's doing. So the basic principle and logic behind them are the same, but it's the kind of like using height instead of weight, and looking at people, or the minimum versus the average. It's about the statistics and the exact things, but the process steps are effectively.
All right.